BASE. Okay. There we go. Okay, so as was said in the introduction, what I'm going to be talking about today isn't, for the most part, the research that I do. The work I do in the laboratory is trying to understand the molecular mechanisms that cause Parkinson's disease. But a lot of the work that I do is based upon the understanding of human genetics, which has come through the huge efforts to sequence the human genome and understand how our DNA can influence not just human disease, but also all sorts of human phenotypes, human characteristics, and so on and so forth. So what I'm going to be talking about over the next sort of 30, 40 minutes or so, and I'll try to leave time at the end for a few questions, is what were the technological advances that have allowed us to gain this huge insight into the human genome? And what are the sort of interesting areas of research in terms of developing treatments for human disease, but also about how we consider ourselves as human, with a particular focus on issues around neuroscience and neurogenetics. And what all of this really goes back to is the work back in the 19th century, which I'm sure all of you are very familiar with, by Charles Darwin and Gregor Mendel, who independently and working from very, very, very different angles, were really critical in terms of beginning to understand why organisms behave and look like each other if they're closely related. Obviously, Charles Darwin was doing this through his work on evolution and the origin of species, and Gregor Mendel was looking at it in a much more mechanistic way, trying to understand in a very reductive system, looking at pea plants, trying to understand the patterns of inheritance. And it was really the bringing together of these two areas of research that allowed us to start thinking about what actually drives that the way in which traits are inherited and passed on from generation to generation. And of course, one of the great things about this is it's something that all of us can relate to, because we all have family members that we can relate to. This is a picture of me back in 1978, sitting on my grandfather's knee. This is a picture of my daughter, who's now 20 months old, sitting on my knee. And immediately you can see that there are shared facial characteristics between us. We look similar. And so this is something that certainly undergraduate students that I talk to here and at UCL, but of course also students at schools can relate to. They can see that family members or related family members look like them. And that's a very easy route into understanding or getting interest in genetics and what that does in terms of driving our understanding of humanity. Now, what really sort of supercharged our ability to understand genetics and inheritance were the really quite amazing insights provided by work in the late 1940s and 1950s by Crick and Watson, shown here in their lab at the Cavendish, and by Rosalind Franklin and Maurice Wilkins and their groups down at King's College and latterly at Birkbeck College down in London. And together their work from a structural point of view, in terms of understanding the actual structure of what we now know is the heritable material, deoxyribose nucleic acid, and at a theoretical level with the work of Crick and Watson, it actually allowed us to get an insight into the structure of the heritable material, DNA, and then get an insight into the actual mechanisms whereby that DNA could pass on information from generation to generation. Now, obviously, the actual elucidation of this took many, many decades, and Crick in particular was involved in understanding a lot more about the patterns of inheritance and the molecular basis of that, especially through his work to generate the central dogma of molecular biology. But really, it was the original insight that the structure of DNA basically facilitated the ability of information to be passed on from generation to generation, upon which all of our subsequent understandings of human genetics really have flowed from. And of course, what it boils down to is the information which is kept within our chromosomes. And this is the human karyotype showing the 23 human chromosomes. For many years, I gave this talk, and I grabbed a picture of a set of chromosomes off the internet, and I only realized a year or two after I started giving this similar talks to this, that actually what I was using was, I think it was an orangutan's genome, because there were too many chromosomes. And I sort of went through, I was saying, oh, if you look at chromosome 12, and I started counting the chromosomes and realized that there were too many. Fortunately, this definitely is the human karyotype, and I double-checked that. And if you zoom in on each of those chromosomes, you can see that each one is a macromolecule. It's a long-chain polymer of the chemicals that Watson, Crick, Rosalind Franklin, and Maurice Wilkins described back in the 1950s. And this beautiful molecule, this double helix with the antiparallel chains, made up of all of these individual base pairs, which is where the information is contained. And this is really what drives our understanding of human genetics. And subsequent to that, there was a huge amount of work defining individual genes, the structure of the human of individual genes, both in prokaryotic organisms and in viruses, where a lot of the early work was done, and then moving into mammalian cells and latterly into humans. But again, sort of thinking about the technological advances and the scientific insights that have really generated a step change in the way in which we understand human genetics, you need to move forward into the 1970s. And the work of Fred Sanger, again at the University of Cambridge, to develop a technique which actually allowed you to read that DNA. So back in the 1950s, Crick and Watson developed their theoretical structure based on the work of Wilkins and Franklin. But we didn't really have an easy way to look at what the exact sequence of that information was. This is where Fred Sanger came in. And incidentally, Fred Sanger is one of the very few people to have won 2 Nobel Prizes, and even rarer, 2 Nobel Prizes in the sciences. So he won a Nobel Prize for his work on the structural biology of proteins. And then won a Nobel Prize for his derivation of what is now called Sanger sequencing, which is really the gold standard in terms of reading the DNA of any organism. This is one of the images from one of his early papers in the 1970s, where he used a technique called dideoxy terminal sequencing, which basically stops the replication of DNA and allows you to create a ladder which you can then read and figure out the actual sequence of the DNA bases in a gene. This was actually from a virus. So originally he worked in very simple organisms with very small genomes. And it was only many years later that we were able to scale that up and understand what mammalian genomes and, of course, the human genome looked like. But all of the information that came through and really filled out our understanding of the human genome came from this same technique. And in clinical applications today, I have an honorary position over at UCL, attached to one of the hospitals at UCL, where they carry out genetic screening for neurological disease. And the gold standard, the one which is approved by the NHS for diagnosis of genetic disease, is exactly the same technique that Fred Sanger developed back in the 1970s. So we have lots of other technology which we can now use on a much larger scale. But if you want something which is a clinical gold standard diagnosis, we still use the technique that Fred Sanger used. And really, it's difficult to calculate exactly how big a debt we owe to the development of this technique and the work of Fred Sanger. And of course, he was honoured by the naming of the Sanger Genome campus by the Wellcome Trust up in Cambridge. So that was back in the 1970s, and it wasn't until the early 2000s that we actually got to the point where we could sequence the whole of the human genome. Human genome is, give or take, about 3 billion base pairs of DNA, and there was a huge effort, a worldwide effort, coordinated by national governments initially and then moving into the private sector to try and get a full readout of what the human genome looked like. Prior to this, and again starting with the work of Fred Sanger, it had been possible to sequence the DNA of smaller organisms and very simple nucleic acid-based organisms, so thinking about bacteria, thinking about viruses. But taking on something the scope of the human genome up until the 1990s really hadn't been something which had been thought to be achievable. And it took a vast effort with hundreds of millions of dollars and pounds and yens being put into this to allow the sequencing of the human genome. And as I say, initially it started out as a public sphere effort coordinated by national governments around the world with particular input from the American government, the British government, and the Japanese government. And that resulted in one publication in Nature, which was the initial sequencing and analysis of the human genome. As you can see, the list of authors on papers, and this is true of a lot of genetics and genomics papers coming through to the present day, are vast collaborations. This involved multiple groups, multiple researchers, multiple funding bodies, a very, very complex series of experiments trying to read the human genome. And most of the information that was included in this initial draft from the public effort was actually developed using the Sanger sequencing method. So it was using a step-by-step sequencing of the human DNA. Now, in parallel to this, and published in the same week, was a paper in the journal Science by Craig Venter and colleagues. And Craig Venter had initially been working as part of the public effort to sequence the human genome, but had got frustrated with the speed at which it was progressing. And so he set up his own company. He got investment from capital investors who were interested in generating profit from the readouts of the human genome and the understanding that that would provide us, and sped up things by developing a shotgun sequencing technique, which allows you to, I won't go into the technical details, but it basically speeds up the rate at which you can carry out sequencing. And so he sequenced, again with a large group of collaborators, a different copy of the human genome. This is where it gets a bit more interesting, and I titled this talk My Genome and Me, because actually this is something which is truly pertinent to this initial paper from Craig Venter, because it turns out that actually it was his genome. So it was DNA that he had isolated from himself, and he was carrying out all these efforts to sequence a genome, but it was actually his genome. And this is, so right from the very start, from the initial sequencing of the first couple of human genomes, there were already implications for personalized genomics and our understanding of ourselves based upon our genetics. So I'm going to skip through some of the interesting things that have come out of genomics, because obviously since this initial sequencing efforts, we now have many thousands of human genomes that have been fully sequenced. And they give us a really unique insight into both human disease genetics, which is where my main interest comes, but also lots of other aspects of human biology and actually how humans developed. One of the more interesting aspects of this, and we were discussing this just before the talk, is that by looking at the span of human genetic variation, so looking in Africa, across Europe, over to Asia, over to America, and to the Pacific, you can actually follow patterns of human migration based upon the genetic signatures which are in the genomes of individuals that are being sequenced during the present day. So there's a couple of interesting things that come out of that. First of all, the greatest degree of genetic variation in the world is around here, in East Africa and Southern Africa, because that was where there was the largest genetic diversity back when Homo sapiens was initially evolving. From there have been sort of bottlenecks at each point, going into the Middle East, then through Europe, over into Asia, over to America, that have reduced genetic variability. So if you go right down to the bottom of South America, which is one of the last places to be settled by human beings, actually the genetic variability in the native population in South America is one of the least diverse in a broad sense across the human populations, where you have larger populations. So I'm not talking about areas where there has been genetic pressure since then. One of the other fascinating things, and this is really something that's only become apparent in the last probably 5 or so years, is that there are signatures in the human genome, and in particular in European populations, of early interbreeding between humans and Neanderthals. And you can actually pick this out in human genes. If you have a Northern European genome, which I have, my ancestors are from Ireland and from Scandinavia, I will carry certain genes where most of that genetic information actually came from Neanderthals. So we have very good genetic proof that there was interbreeding between Neanderthals and humans. And this has only come about because actually we are now able to apply that same genetic sequencing approach that we use to modern day humans to samples fossil samples where we're able to extract enough DNA from Neanderthal bones to actually read a Neanderthal's genome. And this gives you an insight as to exactly how powerful these techniques can be. And it's important to remember that we are still really in the infancy of applying genetic and genomic technologies. The ability to read a human genome has only really been around for 15 years. And that initial sequencing of the human genome was a vast effort. Now it's much easier, and I'll talk a little bit about that in the coming slides. But we are still very much in the early days of understanding human genetic variability and human genetic diversity. One of the ways in which this has happened is through the 1,000 Genomes Project Consortium. And again, this was a sort of follow-up from the Human Genome Sequencing Consortium, which has set out to sequence not just one genome, but 1,000 genomes, and start doing that comparison between individual populations around the world. We now have in the UK the 100,000 Genomes Project, and the aim is going to be to have a vast database of human genetic information, which will aid our understanding of human genetic diversity, but in particular thinking about clinical applications. Now, I work in the School of Pharmacy, so I lecture to pharmacy students, and one of the things that I am particularly interested in is how does this apply to our understanding of disease? And for many years, certainly over the last 10 years, people have been talking about there's going to be this sort of genomics revolution in the way in which we diagnose and understand disease. And actually, in many ways, that revolution is already here. If you read in the papers or see on the news stories about new cancer treatments, a lot of new cancer treatments are being developed and are being targeted by sequencing the genome of tumor cells taken from individual cancers. What that allows oncologists to do is it allows them to target drug treatments to tumors that they know will respond based upon the genetic information which is in that tumor. So we're already entering into an era of personalized genomic medicine. At the moment, that's relatively limited and is very, very expensive. But it is something that is going to become more and more common and more frequent in normal everyday practice. This is something that the British government in particular has realised. They've set up a public funded body called Genomics England. A number of my colleagues are very closely involved in this to try and make sure, first of all, this information is regulated and is looked after in a way which is appropriate, but also look for the opportunities in terms of improving healthcare within the UK and with implications across the world. And actually, if you look on the internet, if you go to the NHS website, you can download a guide to this called Translational Genomics, which gives you an insight into how the NHS and how health providers in the UK are thinking about what are going to be the next steps in terms of actually using this information in a healthcare setting. So this is a revolution which is only just starting, but it is here already. And of course, there's lots of implications of understanding more about our genomes. Just so happened that Gattaca, a film about the impact of human genetic information and a way in which in a sort of dystopian parallel universe it's used to determine exactly what you end up doing based upon your DNA sequence at birth. And of course twins is perhaps the slightly more light-hearted take on all of this. But actually there's been an interest in how genetics and how the information that we inherit that defines some of our characteristics, how that could impact and how that could be misused and used in the public sphere. And a very good example of this, and one which caused a huge amount of controversy, is the example of babies with three parents, which was something which was flagged up in the press a number of years ago about a particular technology that allows you to replace defective mitochondria in unborn fertilized eggs. So the background to all of this is that there is a group of diseases, mitochondrial diseases, which are inherited through your mitochondrial genome. This comes primarily, almost wholly, from your mother. I gave a talk at the Institute of Neurology a couple of years ago and said you get all of your mitochondrial DNA from your mum. And one of the students came up to me at the end and said, well, actually, there are very, very few examples where you can get a couple of mitochondria coming through from your dad, so you're not 100% correct. So I try to qualify that whenever I talk about it since then. But the vast majority of the DNA in your mitochondria comes from your mum. Now, if there's a mutation in that mitochondrial DNA, that is very, very bad, and it can cause very, very serious disease, especially childhood diseases, such as mitochondrial myopathies, where you have serious muscle wasting and serious muscle problems. If you think about it, that's kind of obvious. The mitochondria are critical to the function of every cell in your body. If you have a defective gene in your mitochondria that affects the functioning of that mitochondria, that is very, very bad for you. Now, because that DNA is slightly separate from the rest of your genome, what has been developed over the course of several decades is a technology to, if you're inheriting defective mitochondria from your mother, there's a way to actually remove those defective mitochondria and replace them with mitochondria from a healthy donor. Now, what that means is that any child which is born from this, and this is all carried out by in vitro fertilization, what that means is that the individual who would be born from that IVF treatment would have DNA from 3 people. They'd have some from their dad, they'd have some from their mum, and they'd also have some from the healthy donor who donated healthy mitochondria with healthy mitochondrial genomes. Hence the somewhat scary title in many ways of babies with 3 parents. This work in the UK has been really led and driven by Doug Turnbull, who's a professor up at Newcastle University. And I was very fortunate to go to a talk by Doug a couple of weeks ago in London, where he was talking about where they've got to in terms of applying this technology, and also in terms of the way in which it is being regulated and scrutinised within the UK. And this was actually a couple of days before the government made a final announcement that they'd actually approved the use of this technology, or one type of this technology, as a treatment, as an experimental treatment for people who suffer from mitochondrial DNA mutations. And it was actually incredibly inspiring. It's one of the best talks that I've been to this year, and actually probably over the past 5 years or so. Because what Doug did was he talked about and had video clips from a number of the patients that he works with. And he's a neurologist. He works with individuals who have these mutations and suffer the consequence of these mutations. And he was incredibly passionate about using this technology to provide individuals who have mitochondrial DNA mutations to have children, because many of the individuals with these mutations they're able to become pregnant, but their children often die very, very early if they've inherited mutations in mitochondrial DNA. And he was very passionate about this. And a few days after he gave that talk, actually, the government announced that after a lengthy, and we're talking between, I think it was 3 or 4 years worth of consultation, of very, very detailed scrutiny of the technology and the ethical and moral issues that surround our ability to manipulate the way in which we reproduce in this fashion, that they had approved this specifically in cases of mitochondrial DNA mutation. One of the other things that comes out of this story is that in many ways this is a horse that has already bolted. The technology is already there, it's been demonstrated in non-human primates, and groups and researchers around the world have started using it. This was a story which was in the papers about 4 months ago now, I think, and it was a group based in New York who carried out exactly this type of treatment. It was actually technically slightly different the way in which they were carrying it out, but they'd carried it out in Mexico because there was no regulation of that particular treatment. So they were allowed to carry out this, apply this technology in a fertilization clinic, but without the regulation that they would have in America or they would have over here in the UK. And so this is technology which can work, and there's actually a baby who's been born to a parent who has a mitochondrial DNA mutation and is apparently healthy, although none of this has been scrutinized in the peer-reviewed literature yet, so we don't know the details of that. But it gives you an idea as to how quickly technology is advancing, and that technology is advancing at a rate which is beyond, to my mind, society's efforts to try and understand that technology and think about the implications, both ethical and moral. So I'll give another example, and this again is very much from my own area of research, so thinking about neurological disease. And it focuses on Huntington's chorea, Huntington's disease, which is a purely genetic disease. It's a disease where you only develop Huntington's disease if you have a mutation in a gene called Huntington. It's a particularly devastating movement and cognitive disorder. You develop a jerky gait. Basically, you lose control of your body. You have uncontrolled, sort of almost like spasms, which is a chorea. It's meant to be very similar to someone dancing, but they have no control over it. You also develop an aggressive and very rapid form of dementia, similar to Alzheimer's, but technically a little bit different. This was described many, many years ago by Samuel Huntington, and we know a lot about the genetics of this. It was actually the first neurological disease where we identified a mutation which causes that disease. So it's one where we actually know quite a lot about the genetics. Now, of course, knowing that there is a particular mutation that you can sequence and you can identify means that with IVF technology, there is a way of removing that particular mutation from a particular family if they want to have children. This technology, this technique, is called pre-implantation genetic diagnosis. Essentially, an individual who carries a mutation in the Huntington gene, where they know that they will develop Huntington's disease in later life, are able to undergo in vitro fertilization procedures, develop a number of individual early-stage embryos. You can take a cell from each of these embryos, sequence the DNA in these, and identify which ones have the mutation and which ones don't. Huntington's disease is an autosomal dominant disease. You only need one mutation for the disease to occur. So you can identify each one of these embryos which has two of the normal copies of the gene. That means that any child that develops from that embryo will not go on to develop Huntington's disease. And there's a way of preventing the transmission of this disease from one generation to the next. And again, this is a technology which is actually been around for a while now. It's been used in the UK for a number of years and is a sort of natural consequence of our ability to carry out in vitro fertilization and our ability to sequence our genomes and sequence individual genes. Again, this is something which is available on the NHS. It's very tightly regulated. And if you go to the Human Embryology and Fertilization website, there's a list of genetic disorders where you can undergo this treatment. Now, one of the complicating factors here is that what I've talked about is applying this to a fatal neurological disease. There's no treatment for Huntington's chorea. There are very, very few drugs that even make any impact on the symptoms. But in principle, you could apply this to any human characteristic. If you wanted a child that had blue eyes rather than brown eyes, you could have a look in their DNA and you'd be able to adjust which embryos you selected to have that particular characteristic. And this raises huge, again, moral and ethical issues. And this was raised quite nicely in a slightly different context by Keir Starmer, who was the former director of public prosecutions. He was talking about it in the context of abortions. And that in UK law at the time, this is a few years ago now, that the law does not prohibit abortions that select the sex of the baby. So you could select whether you have a male or female child, based upon, or you could prevent having a male or female child at a later stage in pregnancy. Now, the implication of pre-implantation genetic sequencing is you can do that at an in vitro stage, and you can select whether you have a male or a female child much, much earlier in the developmental process. This, of course, has huge implications for the way in which we reproduce. Prior to this technology being developed, we did not have a say in which bits of our genome were inherited by our children. Now, technically, we do. And as I say, this is very tightly regulated in the UK. So for the UK, pre-implantation genetic diagnosis is used for a very tightly defined and very tightly licensed series of human genetic disorders, all of which are fatal, and many of them are very early onset in nature. But in principle, you could use this for any characteristic. And actually, if you go over to the US, in vitro fertilization clinics over there, a minority, but a sizable minority of them, will allow you to select male or female embryos based upon in vitro fertilization. So again, this is a technology which is being used, and in terms of selecting whether an embryo is male or female, you're looking at a big chromosomal difference. You're not looking at a single change in their DNA. So technically, that's actually relatively easy. That's something that is well within our technical capabilities. And so again, this raises many, many ethical and moral issues. And again, I think it's a really interesting way to get, certainly this is something I do with the pharmacy students here at Reading, but I go into schools every so often and give talks about my work, and it's a really good way to get students thinking about actually what are the implications of scientific discovery and scientific progress. I'll just run through another example of what our understanding of genetics, some of the implications of that. And I'll talk about pre-symptomatic diagnosis. So what I've talked about up till now is ways in which you can prevent disease going from one generation to the next. The other implication of this increasing understanding of our genetic heritage is that actually you can identify people in the human population who are at greatly enhanced risk of developing a disease long before they develop it. In my own field of Parkinson's disease, we now have a list of somewhere around 12 to 14, depending upon how you categorize them, genes where mutations in those genes will cause you to develop Parkinson's. And we can identify those genes at birth, or as I showed with the IVF story, actually long before birth, if you so wish. And what that means is that there are now people walking around who have mutations but don't know they're going to develop Parkinson's disease in later life. And we have a very good, very public example of this in Sergey Brin, who's one of the co-founders of Google. Now, a few years ago, Sergey Brin's mum developed Parkinson's disease. It so happens that his family background is Ashkenazi Jewish, which is the Jewish populations who went to Europe after the diaspora from the Middle East. And for a variety of reasons, some of which we understand, some of which we don't, there is an increased rate of mutations in a particular gene, actually the gene that I work on, a gene called leucine rich repeat kinase 2, in the Ashkenazi Jewish population. It's much, much higher than other European and Middle Eastern populations. So it also so happened that his wife at the time was a geneticist, and had founded a company called 23andMe, which you may well have come across. So she said to him, look, you're from an Ashkenazi background, your mum's developed Parkinson's disease, you should probably have a look at your the genes that we know are at risk of developing mutations for Parkinson's. And so he got his genes checked out. And it turns out that he has a mutation in leucine rich repeat kinase 2, which increases his risk of developing Parkinson's disease at least tenfold. By the time he's 80, he has a 1 in 10 chance of not developing Parkinson's, much, much higher than the general population. So the likelihood is, the balance of probability, is that Sergey Brin will develop Parkinson's disease as he grows older. Obviously, Sergey Brin, as a co-founder of Google, is one of the wealthiest individuals in North America, and he's in a position to do something about that. So he has donated millions and millions of dollars to research into leucine rich repeat kinase 2, and some of the work in my laboratory is funded through the Michael J. Fox Foundation, an American charity focused on Parkinson's research, to work on trying to understand leucine rich repeat kinase 2, and eventually to try and discover therapies that will hopefully treat not just Sergey Brin, but the other many thousands, many tens of thousands of people who carry mutations in this particular gene. As an aside, this is my old boss at UCL, a guy called John Hardy, who's one of the leading human geneticists, especially in the neurological disease area in the world. And this was him receiving a prize from Sergey Brin, one of the breakthrough prizes, which you may have come across. They're sort of the San Francisco Bay Area tech version of the Nobel Prizes. You get a $3 million prize for winning them. This is John receiving a breakthrough prize last year, or sorry, the year before last, for his work on the genetics of Parkinson's disease. So sort of moving along a little bit and thinking a bit more about how personalized genomics is really moving into the public sphere and also the private sphere. 23andMe is a really good example of this. So 23andMe was founded as a personalized genomics company a number of years ago now, and it's a very simple premise. Basically what they do is they take a sample of your DNA and your spit, you send it off to California, they analyze your genome, and they send your results back as in a website form. It's relatively cheap. It's £125 to have your genome analysed in this way. You can do this over the internet, or if you want, you can go down to Superdrug and buy a kit over the counter. There is very little regulation of this, and certainly no clinical regulation of the information that comes back. And there are some very interesting disclaimers when you go through this website as to what happens to your information and the rights that 23andMe have in terms of analysing that information and selling it on to other companies. So again, it's thinking about, do we actually own the genetic information that we are made up of, and what happens to that information? The technology is actually a little bit different to Sanger sequencing. It uses an approach called microarray analysis. Basically, I'm not going to go through this in too much detail, but you have a microchip with many thousands, actually now many hundreds of thousands of individual DNA probes that match up to individual small changes in our genome. If you have a particular variant of a particular gene, the DNA on the chip will react to it and will emit some light, which you can read on a computer, basically. So it gives you a broad readout of your genome. 23andMe does not offer genome sequencing. It offers a sort of blurry overview of your genome with lots of detail on individual variants. But it does actually give you some kind of interesting information. First of all, and going back to what I'd been talking about earlier in terms of following where people have come from and how human populations have migrated, it gives you a readout of where your DNA is from. So you get an idea as to how much is from Asia, how much is from North America, and so on and so forth. But it also gives you a lot of information relating to disease risk. So these are actual snapshots from the 23andMe website. They're actually posted by people with their names included on this. I've blanked those out because one of the things that, as a researcher who works with patient populations, I'm acutely aware of how valuable and how important a person's individual genetic information is. But this is part of the revolution, which is certainly not being tightly regulated across the world. So it gives you an idea as to what your risk is and what the average risk is based upon your genetics. So this doesn't take into account environmental and dietary and lifestyle issues, but it deals with your underlying genetic risk. They also look at a range of inherited conditions. These include a number of different disorders, including cancer, the BRCA1 variant, which it predisposes to cancer, and also the variant that Sergey Brin has. So you can find out, by sending your DNA over to California, whether you have the variant that will almost certainly cause Sergey Brin to develop Parkinson's disease. All of this without any clinical input, without any genetic counselling, without any discussion of the implications of the information which you are discovering. And especially for Parkinson's disease, we have symptomatic treatments for Parkinson's, but we have no way to stop or slow down the disease. So you're finding out information which has huge implications for how the rest of your life will be lived, but without any way to intervene and change that. And this is really quite shocking information. Going back to the Huntington's story, a number of people who are at risk of developing Huntington's are given the option of finding out whether they have the mutation or not, and they very deliberately make a choice not to find out because they do not want that hanging over them for the rest of their life. And so the implications of some of the information you can find out from your genome really can be life-changing and really quite a shock. Just briefly, I'm going to touch very, very briefly on how this impacts on my work. What we can use genomics to do in terms of a clinical setting, in terms of understanding human disease, is we can use it to identify novel variants, novel changes in the human genome that predispose to different disorders. I'm particularly interested in Parkinson's. What we would do is we'd take a large group of patients, and we're talking many thousands of patients. The most recent study of this nature, it's called a genome-wide association study, includes something towards 20,000 Parkinson's patients and the DNA from those patients, and a much, much larger range of human controls. We isolate DNA from these individuals and carry out analysis using this microarray technology. And actually now we're starting to use DNA sequencing and sequencing the whole genome. It's got to the point where if you have the right machine, you can sequence the human genome for under $1,000. You need a £25 million machine, but the actual sequencing is about $1,000. And as time goes by, it will become relatively easy and relatively cheap to sequence your entire genome. I can certainly see a point not in the too far distant future where when you are born, instead of having a little sort of pinprick test to test whether you have a blood disorder, you have a sample of your blood taken as an infant, and your genome is sequenced, and that stays on your NHS records for the rest of your life. That is not too far away, and is actually not going to be all that costly. What we can do as researchers, so stepping outside of many of the implications more generally about this, is we can look at the human genome, and this is a representation of the human genome in the round, if you like. So these are each block is an individual human chromosome. We can identify changes in your genome which are more common in people who have the disease than those who do not. And that gives us an insight from from the molecular side of things as to which genes are involved in risk of developing disorders. Basically, each bit of the human genome, each of these individual dots is an individual genetic variant in the human genome. Every red dot in this circle that sticks out from the human genome is a bit of the genome which is significantly associated with Parkinson's disease, tagging a particular bit of a genome, and in some cases, individual genes, and giving us an insight as to what genes we can target in terms of drug development and drug therapy for Parkinson's. And this is work that I do. I work on these genes, trying to find out what they do, and trying to develop treatments which will alter how they behave in Parkinson's. The idea is that we can develop drugs which will slow down or stop the development of Parkinson's disease, both in people like Sergey Brin who have an inherited form, but also in the more general population who do not have an inherited form of the disease. Just very, very briefly, and sort of touching on the wider implications of this again, one of the areas which has been controversial not just for the last 10 years, but for the last 150 years, is the impact of inheritance and heritability on intelligence. And of course, in the context of schools, what this means in terms of pupils, both in terms of their ability and the way in which they are taught. This came into the public domain quite violently, I would say, a couple of years ago, where one of the aides to Michael Gove was very, very forthright, a guy called Dominic Cummings, very forthright in saying that basically we should be streaming students when they get into schools based upon their genetic background. And you can predict how well they will do at school based upon their genetics. And this goes back to a much, much longer controversy. And this is Francis Galton, who was a follower of Darwin, but was also the founder of eugenics, the scientific discipline looking to basically modify human evolution and improve that evolution. And obviously this had many disastrous consequences at a global level, in particular if you think of the impact of the Holocaust during the Second World War, and was very, very strongly discredited over a number of years. This is a book he wrote back in the 19th century called Hereditary Genius, where he followed, to his mind, the inheritance of genius in families. All of the geniuses, or genii, I don't know what you would call them, all of them were male, by the way, so it was a sort of male-dominated inheritance. And actually, when you read it, and you can actually download this off the internet, a scan of the original book, from a modern genetics point of view, it is incredibly naive in terms of how we understand the interaction between your environment and your genetics. And when you look into the cutting-edge genetic analysis of intelligence, which actually Dominic Cummings was referring to, he talked to a human geneticist who works on intelligence, actually the evidence is that there is an impact of your genetic background upon IQ, which is a very crude measure of intelligence more broadly, so there are a huge number of caveats to this. But that actually it's a very, very good example of incredibly polygenic inheritance. So what it means is you don't have one variant in one gene that means that you're going to win a Nobel Prize in later life. What you have are a very large number, probably into the hundreds if not thousands of genetic variants, which have a tiny impact on your ability, your cognitive ability at a fundamental level. So taking out any impact of environment or diet or so on and so forth. And actually, this is a similar representation to the one I showed you of Parkinson's disease, where the red bits were the ones which were significant across the genome for inheritance or inheritance of genetic risk for Parkinson's. Here, you've got the same plot, a slightly different representation. It's a sort of linear one. These are individual chromosomes. And any genetic variant which is significant across the genome for the heritability of IQ would have to go past this line. This is a statistical boundary over which you have to get for it to be considered significant. This is one of the most recent genome-wide association studies for IQ, so I'm separating that out a little bit from intelligence because they are not the same thing. And none of them reach significance. What that means is that no one single variant has been demonstrated in a genome-wide population-based study to directly influence your IQ score. That's a very important demonstration, and one which has a huge impact in terms of the idea that we could sequence the DNA of somebody before they go to school and say, you go in one class, you go in another. That is quite honestly, it's a fantasy, and would be very, very dangerous from my point of view. But there are people out there in very privileged positions of influence who are arguing that right now. So again, this is an example of the potential societal impacts of, I would say, misuse of genetic information. Others would say this is a way we can use it to improve the way in which we educate people. I'll leave that for you to make up your own minds. And very, very finally, just for the last few slides, of course, we're now in a position to actually change our genomes. So over the past 5 to 10 years, genome editing technology has rapidly made progress. In particular, an approach called Cas9, which was developed partly from bacteria, partly from plants, which is a way of changing individual base pairs in an organism's genome. This was originally demonstrated in bacteria and plants, but we can now use in a recombinant fashion, so we can purify the individual bits of the machinery that does this. We can use this technology to alter human genomes. And again, this is already being done. There's a researcher at the Francis Crick Institute, which is next to St Pancras Station in London, who has been licensed in the UK to edit human embryos to try and understand human development. And so this is a technology that allows us not just to read our genome, but to change it. And the implications of that, given all that I've talked about before, is clearly incredibly, it's a change in the way in which humans interact with life, basically. Again, very briefly, and I will only mention these in passing, this is something which governments are taking stock of. They are trying to regulate and to provide a structure within which this kind of information operates. A good example of this is health insurance. Should health insurers change the way in which they price your health insurance based upon your genetic background? In America, that was legislated against several years ago now, back in 2003, and it is illegal for a health insurer over in America to basically discriminate against you based upon a mutation you have in your genetic background, which will either predispose you to a condition or cause you to develop a condition. In the UK, obviously we don't have the same kind of health insurance system, but there is a much, much weaker piece of, it's not even legislation, it's a sort of agreement between friends, basically, which says that insurance companies won't use genetics as a tool to alter their view of risk for you. There's a huge amount of information about this on the internet. I particularly recommend the Nuffield Council on Bioethics, who have very, very detailed and fascinating discursive documents about the implications of genetics and neuroscience for all many, many areas of human society. And as I say, this is very, very tightly regulated, especially the embryology side of this, in the UK by the Human Fertilisation and Embryology Authority. And again, they have a lot of information on their website about genetics, human inheritance, human disease, and so on and so forth. I realise I've only left you 10 minutes to get to your next place, but I'm more than happy to take any questions if anyone has any. Thank you very much.